So there are some differences between the productivity cycles at the temperate area and near the equator and near the poles. So first we'll look at the tropics, the area near the equator. Here, there's much less seasonality. It's pretty much just warm and sunny all the time. If you recall back to like how seasons work. So it's pretty much just warm and sunny all the time. So there's lots of light year round. And yeah, not much seasonality. So that means productivity doesn't change much. There's always plenty of light. And so there's just kind of a constant level of nutrient limitation. So primary productivity kind of goes up and down with how much nutrients are around, but it's pretty steady throughout the entire year. So there's not those big peaks and valleys that we see in the temperate. Now at the poles, there's enhanced seasonality. So the seasonality is more extreme, right? For example, in the wintertime, if you're close enough to the poles, there's times of year where the sun like doesn't come up, like where it's just dark for 24 hours. And during the summer, there's times where it's light for 24 hours. So that means that in the winter, there's very little light, and pretty much there's extra light in the summer. So summertime's really the only time we're going to have like enough light for photosynthesis. And so instead of getting those two peaks, you just get one big summer peak. Because again, there's a lot of light in the summer and then like not much light in the other seasons. So you just get one big summer peak when there's a whole bunch of sunlight in the summer. And the other times of year, it's pretty light limited. So it looks a little like this. Last thing I'm going to talk about for this lecture is how we study plankton. There's two main methods. There's some others as well, but these are the two main ones that I know of. Plankton nets. This is like what I used to get our plankton sample for our lab last week. It is a net with various you can have different mesh sizes. Some of the meshes, they'll be bigger. Some of them will be like really, really small, so small it kind of looks like it's solid. And they will collect any plankton that are, sorry, not smaller, larger than the mesh size. Sorry, that's a typo. Anything that's larger than the mesh size will stay in the net and will end up in your little cod end. And then you take that plankton sample and you bring it back to the lab and you dump it out and you count and count and count and count your plankton in order to get an idea of how much is out there. So the downside of this piece of equipment is that it doesn't really work for bacteria or viruses because they're so small that you can make a net that has a mesh that small, but it's gonna like really, really suck to pull through the water because like the mesh is so small. So it's not a great way of capturing bacteria and viruses. But the plus side is that you can get quantitative data. So you can calculate density. You can calculate how much water went through the net and like how many copepods did we get in that water. And you can calculate, okay, what's the density of copepods in that area? So you can get this very quantitative data. Another way that you can study plankton is by looking at the DNA that plankton leave behind in the water. Because when plankton are out and they're living and they're shedding stuff, they're shedding DNA all the time, not just plankton, but actually like all basically organisms in the ocean are constantly like shedding DNA, cells, etc., into the water. And so you can actually like take a sample of water and just sequence all the DNA in that water to get an idea of like everything that's out there. And I've been involved actually in study projects that have done both these things. I have both counted zooplankton under a microscope, and I've been involved in a project that was sequencing DNA as well. So the typical way you do this are through things like eDNA and genomics. If you want to learn more about those, that's kind of what it's called. And this is how we find a lot of the bacteria and viruses, is we basically find their DNA sequences and we're like, oh, hey, look, there was like a bunch of different, bacteria in this water, a bunch of different viruses in this water. However, this typically, there are some methods out there that try to get more quantitative with this, but typically it's mostly what we call presence-absence data. So for example, let's say you find Staphylococcus DNA in the water. All you kind of know is, okay, Staphylococcus is like in this area close to when we took the sample because DNA degrades like pretty rapidly. So you kind of know like, okay, like within about a day of when we took that sample, Staphylococcus was like in this area. But you don't know like how much Staphylococcus was there. You'd have to take multiple samples if you want to know like at other times if it was there. So it mostly gives what we call presence absence data, where you just either know it was there or it probably wasn't. So really good for getting at viruses and bacteria, but usually less quantitative. Again, I know there's some development of techniques for making this more quantitative. Uh, and some people actually do things where they'll like add fluorescent dyes and try to like actually count cells and stuff like that for bacteria and viruses. I know there's some of that out there. But these are the two methods that I'm most familiar with for studying plankton. So with that, that is it for this week. Here's the study guide for this lecture at the end. I hope you all have a lovely week, and I will see you on Friday. Where's that? There it goes.